So my name is Chester Chamberlain. I'm coming to you here from UCSF, where I'm a faculty member in the Diabetes Center. First, I'd like to thank Bina for giving me the opportunity to speak today about our work and our ongoing collaboration with them using their fantastic bioinformatics platform. We have a lot of exciting projects going on at UCSF. We're in a rather unique position where we have an exceedingly large number of patient samples, interesting patient cases, lots of collaborations, international collaborations, China, other parts of the United States, Europe, where we're actually bringing in very interesting families with people with diabetes, families with histories of cancer, and we're in the unusual position of just having way too much sample and not enough information that we can get from those samples. This has really been a big bottleneck for us. And so Bina has really stepped into that void and has provided us with a really powerful solution for our projects. What I'm going to be talking about today is just a very small sliver, one aspect of one project that's ongoing right now. So the kind of cancer that we like to focus on is called pancreatic neuroendocrine tumors. And so pancreatic neuroendocrine tumors, are more commonly called PNET, are a very rare form, but a very deadly form of pancreatic cancer. Okay? And this is the kind of cancer that arises from the endocrine islets within the pancreas. For those of you who are not familiar, the endocrine islets are a very small subpopulation within the pancreas. These are the hormone-producing cells. They make up about 2% of the entire pancreatic tissue within the pancreas itself. Okay? This is not a very well-known disease, pancreatic neuroendocrine tumors. Its cousin, which is much more well-known, is pancreatic cancer, which actually derives from the exocrine pancreas, which makes up 98% of the pancreas instead. PNETs are far less known, but they do have a very important role in human health, in particular in health in the US, because as exemplified here with this picture with Steve Jobs, he unfortunately fell victim to a pancreatic neuroendocrine tumor. Okay? And so he actually had an insulinoma, which is a tumor that derives from the insulin-producing beta cell within the pancreas. So this is a very real disease. It's a very rare disease, but it is a very deadly and a very real disease that impacts real people. But again, it's very rare. The incidence of this kind of cancer is about 1 in every 100,000 people. One of the issues with this cancer is that it's actually very slow growing, and when it does grow, it grows very silently. So people with these kinds of tumors actually don't present with any kind of symptoms. The consequence is that 65 of the patients who actually come into the clinic and are diagnosed to have a pancreatic neuroendocrine tumor have already reached a stage where the tumor is no longer resectable. And the only options then are somehow to be able to treat it globally with a drug, for example, which there aren't many available, and none of which are actually effective. One of the big challenges about pancreatic neuroendocrine tumors is they're very unique as well. And so many of the genetic alterations that occur in pancreatic neuroendocrine tumors, the alterations, the variants that are present that actually drive the growth and survival of these kinds of tumors, are not things that can be directly targeted. The variants that we do tend to find are things that are in what we call tumor suppressors. And tumor suppressors are often not enzymes, so they're not easy drug targets. And in addition to that, many of these tumor suppressors are simply not expressed. And so the fact that they're not expressed allows the tumor to grow and develop, and so of course you can't target something that's not there. A solution to that is to identify things, or to identify what we typically call synthetic lethal interactions. Okay, so just a little quick background here. The talk's very short, but I'll go through this briefly and quickly through these different concepts. Okay, but the concept of synthetic lethal interaction first arose in yeast genetics several decades ago. And just to outline that here, if we focus on two genes, we can call them A and B. What happens is that when we inactivate one of these genes, A, for example, the cells remain viable. There's no effect. Or if we inactivate another gene called B, there's also no effect and the cells remain viable. However, the combination of the two, where both are inactivated, results in death. This is the concept of synthetic lethality. This concept has been adopted in the cancer field for several years now, and as it applies to pancreatic neuroendocrine tumors, we can think of it this way, where the A gene, for example, is something that's unique to the pancreatic neuroendocrine tumors, a PNET variant, for example, a variant in a tumor suppressor that's unique to pancreatic neuroendocrine tumors. And B is a drug target, something that we can already target with existing drugs or a drug that could be developed to hit B. And in both cases, so when you have this PNET variant here, it either drives abnormal growth or there's no effect on the endocrine cell itself. And in the same situation here, in the presence of this drug, when you're inhibiting B, you have no effect as well, but the combination of the two results in death. And so this is very useful because you can imagine a drug, for example, having no effect on normal endocrine cells because you're only inactivating B here, but you'll specifically target pancreatic neuroendocrine tumors, which have a unique characteristic A. So it's a combination. How do I identify these kinds of synthetic lethal interactions? Well, again, using the Bina platform, we have large samples, large collections of samples of pancreatic neuroendocrine tumors. And what we and several others have been doing exome capture from these kinds of tumors. And using the Bina platform, in particular the RAVE module, we're able to take this whole exome sequence capture and identify variants that are present within these tumors, and use another module called the AIM module to identify disease variants from those variants, and basically ask the question, what does the landscape look like within these kinds of tumors? What are the pathways that are being activated or inactivated? And one of the striking things that have come from this analysis, and again, it was us and several other people as well that have confirmed this, is that there's a very common, very powerful oncogene called KRAS. And this is present in a large number of tumors and is a very active driver of tumor growth. KRAS is one of the biggest targets in the cancer field today. There's a striking absence of activating KRAS mutations within pancreatic neuroendocrine tumors. This pathway doesn't play a role in this kind of cancer. Instead, what we find is that there's variants or mutations, inactivating mutations, in tumor suppressors, in particular the MEN1 gene, which occurs in about 50% of sporadic pancreatic neuroendocrine tumors that we find in patients. Having learned this information, we performed functional studies, and we tested hypotheses, being guided from the information that we got here from the NGS analysis, and we were able to build a novel kind of pathway, in particular a novel interaction between the RAS-MAP kinase pathway and the MEN tumor suppressor, where the MEN1 tumor suppressor normally blocks the ability of this pathway, of the MAP kinase pathway, from driving growth and survival in endocrine cells. But in tumors, where MEN1 is now gone because it's been mutated, this pathway now dominates the growth and survival of pancreatic neuroendocrine tumors. And to illustrate that with an experiment, one of many experiments that we did is, here we go again with a normal set of endocrine cells. This is a primary islet culture that you're looking at here, and we can culture these, and the white spots tell you these are proliferating cells within proliferating endocrine cells within the islet. When we inactivate MEN1, specifically within the endocrine cells, we get hyperproliferation, transforming these cells into a neuroendocrine tumor cell type here. If we treat these endocrine cells with a MEK1/2 inhibitor, in this case Mekinist, which is developed by Novartis and GSK, we find that there's no effect on the viability of these cells. However, when we combine MEN1 loss, which again is a unique feature of PNETs, with MAP kinase inhibition through the MEK1/2 inhibitor, we get synthetic lethality, where more than 80% of the cells will actually drop dead. So for us, this is a very novel and interesting finding, and resulted from our knowledge that we could glean from the NGS analysis, coupled to follow-up functional analyses as well. So we had this information. What are we going to do with it? Well, we're very excited about it because we think we may have stumbled upon yet a new treatment that could be very effective for people who have these kinds of cancers or tumors. And so what we're doing is we're moving ahead with preclinical trials. We're very fortunate at UCSF where we have what is perhaps the only available patient-derived xenograft model for PNET that's available for our use. And basically what a patient-derived xenograft is, for those who don't know, is that from a patient who had a pancreatic neuroendocrine tumor, in this case actually an insulinoma, this insulinoma was extracted from this patient and then inserted into mice that have been immunosuppressed and then propagated within these animals. These animals can then be tested and treated with a variety of drugs, and in this case we're going to be testing Mekinist, and see what the effect is on the growth and the survival of this xenograft pancreatic neuroendocrine tumor, and see if we can build a case to support future human trials from data we can collect from this model. One of the questions that immediately comes out at us is, well, gee, how representative is this model, and can we predict whether or not we would expect inhibiting the MAP kinase pathway would actually have an effect? To answer this question, we again turn to Bina and their platform, and we perform exome capture from the xenograft itself. Again, employing their modules, RAVE to identify the variants, and then AIM again to identify disease variants. And what we found from this bioinformatic analysis is that the xenograft does indeed contain an MEN1 variant. There is a mutation in MEN1, a frameshift mutation that's predicted to result in a truncated protein within this xenograft model, and therefore make it a null allele. And so we would predict, based on this bioinformatic analysis, that the xenograft would actually be responsive to a treatment, in particular for something that inhibits MEK1/2 or the MAP kinase pathway, something like Mekinist. So I'll just simply say the result. So not only did we identify that this MEN1 mutation is present bioinformatically, but what I did have also was a piece of data showing that we confirmed this experimentally as well. And so we've done immunostaining, Sanger sequencing, and other assays, and we confirmed that not only does this come up bioinformatically from the Bina platform, but we actually look at the primary xenograft tissue compared to other human tissues that we have, and the MEN1 mutation is truly there, and the xenograft doesn't produce it. So we have ongoing studies right now where we're actually treating these animals with the MEK1/2 inhibitor, which is already an FDA-approved drug for melanoma. And we're hoping that we can build an evidence in case where we can actually repurpose this drug, not just for people who have melanoma, but also have pancreatic neuroendocrine tumors. And we've also initiated human trials as well at a very early stage right now, with the idea of coupling both the studies that we're doing preclinically with a xenograft to identify the optimal conditions and the right kind of design trial so we can actually benefit patients as well who have pancreatic neuroendocrine tumors. And so we're coupling both together. The NGS analysis has been very powerful to pretty much validate our PDX model here, but at the same time we can recruit patients based on their MEN1 status, for example, within their tumor. And so this will help us stratify and come up with a very rational design for the clinic as these trials move forward. Okay. And so this was a very short talk. But I'd like to acknowledge all the people who did a lot of work. Mike German, my boss, who is the clinical director at the UCSF Diabetes Center. Eric Nakakura, who generated the patient-derived xenograft in his model, in his lab. Emily Bergslund, who's coordinating the human trials, and a lot of other people who contributed to the work. I'd like to say thank you to Bina for inviting me to speak, but also allowing me to use their platform. It's been really great. And of course, my funding agencies that allow this work to happen as well, the NetRF in particular. Thank you very much.